All right. Good morning, folks. How's everybody doing today? Everybody doing good? Good. Thank you all for attending today's briefing. Today's briefing, we are discussing macular degeneration and some of the advancements in treatments and therapies that are on the forefront of research and what research is doing to be able to help treat patients with AMD. Today's briefing is sponsored by the Alliance for Eye and Vision Research. I'm Dan Ignazewski, and I'm the Executive Director for the Alliance. And we're here, our mission is to for patients and for trying to improve outcomes for patients in the long run. And vision science plays an incredible role in that, and so that's our mission, is to help people understand that value. Excuse me. We want to thank our sponsors and supporters of today's briefing. This includes Research to Prevent Blindness, who provides a grant to help operate and run our research savings site, Restoring Vision Initiative. The AMD Foundation, who is helping sponsor and support today's speaker, as well as the lunch that you'll have in the back of the room. ARVO, the Association for Research in Vision and Ophthalmology. Apellis, who is a manufacturer of pharmaceutical treatments for AMD. Genentech, and Vision Aid, who is in the back, who is providing the headsets where you'll be able to experience at the end of the briefing what AMD patient experience really is like. So a little bit about AMD. Age-Related Macular Degeneration is one of the most common eye diseases in people over 40 in the United States, and it affects the central vision, and it really has a direct impact on patients' ability to live their life independently, live well, and experience the world around them the way that somebody who doesn't have AMD is able to. Over 15 million Americans currently have AMD. It's estimated to double by 2050, primarily due to an aging population. Over 10,000 Americans every day are turning 65 in the United States. So with our baby boomer population aging, we expect the prevalence of AMD and other eye diseases to significantly increase by 2050. It does not cause complete blindness necessarily, but it does affect your central vision, as I mentioned, with the experience goggles in the back. At the end, you'll be able to see some of the different phases of macular degeneration to see sort of what the early stage looks like all the way to some of the late stages. It doesn't necessarily completely blind, but it does cause significant maintain vision you do have. Today's speakers, as I mentioned, are supported in part with our partnership with the American Macular Degeneration Foundation. And we have a leading researcher, Dr. Neena Haider from Harvard Medical School, who will speak about some of the advancements specifically in gene therapy, but sort of broadly around AMD. And then we'll also have a patient perspective from Mark Roser, who is a patient from Connecticut. He's a patient advocate, an engineer, and a researcher himself. So I will turn it over now to Neena. There you go.
Thank you. Good morning, everyone. Very nice. Thanks for coming today. I know there are a lot of young scientists here, and there are people that don't have a science background, so I'm tailoring this to both of you. For the young scientist, the beginning of the talk, I want you to pay attention on how to present science for a non-science audience. So I'm going to give you an outline. I'm going to introduce genetics. So we're all going to get a little bit of genetics 101, a little bit about AMD. I'm going to talk about what's up and coming, so the new trials, and something that came out of my lab too that is just going to start trials very soon. So it's hot off the press. And then we'll have some concluding thoughts. The retina. The retina is the camera of your eye. And it processes in a remarkable way. Vision actually occurs where? We're going to kind of have fun with this. Vision occurs where? Any guess?
Brain.
The brain. Vision occurs in the brain. Light goes through the front of our eye to our front of our eye colleagues, and very important, and then passes to that lens. It has to stay clear and all the way to the back of the eye. And the retina, which 70% is made up of photoreceptors, little signs in there. Those are your rods and cones. They're responsible for your color vision, for your night and day. And then that signal turns from light to electrical signal and gets sent to your brain, and voila, you're seeing all of this in an amazing way without having to think and do anything yourself. When we study genes, or you're studying on a molecular level, whether it's in a dish, or it's in an animal model, or it's from patient samples, here's where we start from. If we think of the entire genome that's in every cell of your body as the world, what we're looking at in a single gene is like the front door, but your bird's eye view, you're sitting outside of Earth, maybe on the moon if you like the moon, and you're looking down and trying to find that front door. That is the level of microscopic detail and complexity that goes into the science and the research that we do. Genetic diseases are categorized in simple and complex. Age-related macular degeneration is a complex disease. Simple diseases have primarily a genetic component. And we're shifting the gears from when I learned Mendelian genetics, and I only look 25, but I'm not. But we had an idea that a simple gene, like in retinitis pigmentosa, one gene mutation and you get the disease, but that's not really the case because the body works in concert with lots of things. Complex diseases also environmental influences. So that's what makes it complex. Genetics and environment. You're a product of your environment. To illustrate that, and I know the middle part looks like a hot mess, I'm going to use my cursor. Can you guys see the cursor? Okay, good. That's going to be my pointer here. So you have genetic perturbations, and something like AMD, we still don't know all the causes of AMD, which baffled me when I first got into the AMD field. What causes AMD, and why don't we know all the causes yet? And how are you going to find treatment without knowing the cause? And then those particular genetic perturbations actually impact networks. And I wrote networks singular, but it really should be plural. Networks and networks of genes. And those network perturbations are what actually impact disease outcome, along with, remember, our complex diseases, our environment. So it's this combination of both that come together in a very dynamic and complex way. And age is one of the phenotype being observed trait that gets you a disease or a clinical outcome. And if you're like me, you don't go to the doctor till you actually feel like something's wrong with you. And even then, you might not go. And so by then, this disease outcome has already far progressed because the perturbations have been happening over time. Genetic terms. DNA, it's your genetic code. It's your blueprint. And I like cooking, so I use cooking analogy. We'll talk about making chocolate chip cookies. Think of DNA like your ingredients. It's in every cell. You got all of it there. You don't need all of it. If I'm making cookies, I only need a few ingredients. I don't need the whole pantry. DNA turns to RNA. RNA turns to protein. Protein are our cookies. RNA is the recipe. If I add a little more vanilla, it's going to change the flavor. If I add walnuts, it'll really change the flavor. And if somebody's allergic to walnuts because they have another genetic perturbation for that, it's not a good thing. See where I'm going with this? Great. So genes are just segments of that DNA. There are about 20,000. Now, I was part of the Genome Project, and when we first started the Genome Project, we thought there were 80 to 100,000 genes. And as we progressed every year, sequencing the human genome, we began to realize, no, there's not 80 to 100,000, there's 50,000. There's not 50,000, there's actually only 20 to 25,000. In your retina, the camera of your eye, there's only 2 to 3,000 being expressed, so about 10% of that genome is expressed there. I'm going to talk a little bit about gene therapy. It's one of the ways that you can hopefully fix the problem that have to do with retinal diseases and macular degeneration. So there are 3 very simple, basic ways to do this. Gene augmentation, which is what all of us probably grew up thinking gene therapy is about, where you put the normal version of the gene in. And usually that's going to work if you don't have a functioning gene. If the gene's not functioning, it's not doing anything, you put the normal version. It's good, because you have to remember dosage does matter. There's gene inhibition therapy. That's the CRISPR. We all heard the buzzword CRISPR a few years ago. It's going to work. It takes out the abnormal. It works if you have like what we call dominant negative. That means there's a mutant gene there and it is functioning, but in a really bad way. And so I'm going to replace it with something good, and that'll work. Well, we don't even know. For RP, there's 250 ways to get this disease. For AMD, I'm telling you, there's probably thousands of iterations, of combinations of genetic perturbations that can get you that disease that are unique to the individuals. How are we going to develop therapies for that? And my focus in my lab has always been genetic modifier therapy. What that does is, in the presence of those genetic perturbations, let's find master regulators. I call them like the circuit breaker of the system. It resets. It resets not one gene, not 10 genes, not one network, hundreds of networks. I love this slide, whether you're in science or not. It's a great reminder for all of us. I'm very happy to see a really diverse group sitting here because we're really pretty much alike. 99% of our genome is pretty similar. And it only takes one little base pair to create this variation, the variation of normal. Normal, there's a wide range of normal, there's a narrow range of disease. And the severity of disease and the disease outcome also alters by those just single base pair changes. So AMD is a complex disease with gene and environment interaction, and I coined this term many years ago. Disease outcome is really determined by a mutational load on the biological system. And that is the primary mutation plus the modifier, plus other factors from the environment that influence epigenetic. And they can influence then how the DNA impacts on networks of genes, not just single gene at a time. I'm going to give you some AMD background now and visuals on what AMD is. So these are from patient samples, and this is just to illustrate the AMD dry and wet. And in the dry form, 90% of patients have dry AMD. They have this drusen deposits. You see here these flecks. And then this area that looks thin here can sometimes break, and that'll turn into geographic atrophy. And wet AMD to date. And not to gross you out, but you'll understand the analogy. If you've had an infection in a cut and you get a little pus, that's exudate. So another visual that shows you histologically too, dry AMD is accumulation of the drusen between the retina and the choroid. So the RP and the choroid, right under that retina, they nourish those cells. Is a problem where the trash doesn't get taken out. That's the analogy I like to say, is your cells process, during that process of turning light into the electrical signal, you get a lot of trash. It doesn't get taken out in this case, and that accumulates. In wet AMD, CNS, the central nervous system, responds to injuries by blueprint of how to make a blood vessel, and they all, whether it's macular, that's the take home on wet AMD. There's another disease, Stargardt disease, and I'm apolitical. I'm sitting in Congress, I'm very apolitical. Stargardt disease, sometimes, some people don't like to think of this as in the AMD category, but you have 2,000 genes in the retina, and I'll tell you from the work we've done, there are similar genes and mechanisms that play in. It's the same network. So I like to bring this in and say, these patients, they have autofluorescence, and they have those little flecks. Now, these are lipofuscin, and they don't clear from the RPE. So when I think of it mechanically, it's the same kind of problem of the trash not getting cleared out. It's the same kind of problem, therefore, mechanistically, whether it hits in the 30s or 40s, and Stargardt can actually hit a little later in life as well, but it's the same kind of problem. And then you have these retina that tear and break and they die off. This is a very simple, very conventional And please go to our friend Taylor, who has a more sophisticated way to show you this information, but the Amsler grid is a vision test. And this is how just a grid, a normal vision versus how it gets distorted. And I tried this last night and I thought it was fantastic, Taylor, and how you understand that it's not actually black and white. There's nothing black and white in the world. That actually the patients are seeing gray tones and gray zones. I think it's really important for us as scientists to also, and as congresspeople, to understand the perspective of the patient. And we're going to have an outstanding speaker who's brilliant at developing these type of tests for self-test. I'm going to segue now for the next few minutes and tell you cool and up-and-coming trials, drugs on the market, and science happening. So I picked this as a hot topic in genetics. It got a little press recently. The main scientist is Dr. Meg DeAngelis, and this is humans genetics. So I developed therapeutics and studied in my lab as a mouse geneticist. But we collaborated very well because I like the balance of understanding what's happening in patients and how can I translate that and understand that, not at end stage of disease, but maybe early, maybe intermediate, how that information comes together. And there's this paper, if you haven't read it and you're studying AMD, take a look at it. It's a systems biology approach. And what it did was it discovered new pathways linked to AMD. I do modeling. So AMD is only naturally occurring in the human species. So when we model the disease, this very complex disease, we're only modeling a little phenotype, one phenotype or two portions of the disease. And that's the take home I want you to get from this slide. I don't expect you to read it. I want you to understand there's a lot of models. Each of them have only one piece of that disease covered. There's a lot of risk factors that go into treating that impact AMD environmentally. They're listed here on this left panel. You can see them. I'm not going to go over them. What I want to talk a little bit about, and I'm going to spend the next few slides telling you what the newest stuff is so when treatments are developed, they're really developed to hit one aspect of those networks or another, maybe one major network, whether it's lipid in the drusen, or it's inflammation, or it's oxidative stress. So the retina is the most metabolic tissue in your body. It processes things all day long so fast, and therefore releases. And I'm sure, even in the, in our general language word, we've heard of antioxidants and how they're so important. So this is a really highly metabolic tissue, and that distress in the system impacts disease as well. And this thing, complement has been around for many, many years. And the big question in the field was, does it play a causal role or is it an indicator? So here are really exciting treatments. They came up with this complement. So they hit the complement from that network I showed you in all the different aspects. They hit the complement pathway, and they have an inhibitor for that. And they've done intravitreal injections. They've done a study. These are all from the presented this at ARVO, so I'm presenting to you what's the public information. Intravitreal injections, so that's great, not subretinal, it's less invasive. Monthly and every other month to treat geographic atrophy, which is when the retina is so bad that it's just going to tear and there's a hole in there, to dry AMD. And they saw 29% reduction in the geographic growth rate. So that means there's less rate of it growing and breaking up, and therefore improved vision. The next drug on the market, and this is what's exciting in the field, these are on the market now, is Izervay, and there's another name for this, and this is by Astellas. Same complement pathway, but a different inhibitor. And these are also exciting. The young emerging scientist, speaking to you for a second, to be in this age now where the genome's done. We know a lot about mechanisms. We know a lot about networks. You get to help develop the next set of therapies. It's cool. Now I'm going to tell you about what this was my takeaway from ARVO and AMD particularly. There are 5 new phase 1, phase 2 clinical trials and 4 new phase 3 trials. So there's 9 trials happening right now in this space. 2 trials are for wet and dry AMD to tackle the diseases at the whole. 7 for wet AMD. 4 trials, which is by the Pulsar group that was just on aflibercept. And these are, and then 5 novel treatments that are up and coming trials. So hopefully will excite you. There's one on CD59, which also hits that complement pathway. And then from NIH, Kapil Bharti's work, I don't know if you all, if you're not familiar, get familiar with his work. It's really cool. Using iPS cells, so these pluripotent cells, to, in a dish, biologically make some RPE patches and then transplant them. Then there's other trials on treatments that are getting attention now on inhibitors for wet AMD. And then there's a nanoparticle and something with an angiogenic. That means anti-vessels. So it's going to tackle that part of wet AMD. Now I'm going to tell you a little bit of my science and a model that we have. This is a mouse model, the only that I know of, multigenic genetic model. So remember when I said we're modeling, we have pieces of the disease. So this one has a really cool part that it's the only one that has multiple genetic perturbations. I thought this is a golden egg here. You have, when we discovered this a few years ago, and when we mapped this, and this paper is in review currently, we found 4 different loci, regions of the genetic genome where there are perturbations associated with the disease, and those regions all have AMD genes. This is another cool aspect of this model. It not only has the dry phenotype, it has the wet phenotype. It's like checking all my boxes of this is a good one for me to look at, to study, to understand how does this disease happen, what is the mechanisms underlying, and how can we develop treatments for this. Third checkbox, or fourth, was just like in patients. Sometimes the disease is mild, sometimes it's severe, sometimes it's unilateral, sometimes it's bilateral. So this, to me, was one of the most outstanding models that I have seen come around from a geneticist perspective on AMD. Now, one of the wheelhouse of genes that I look at, they're called nuclear hormone receptors. I like nuclear hormone receptors because there's 52 of them. They regulate everything in your body. Most of them are ubiquitous express. This one in particular regulates all of these networks and a lot more. I can't fit them all in a slide, but just to give you an idea, it regulates neuroprotection. So how can we protect? Version of light energy to electrical signal. It regulates that oxidative stress. So maybe, and lipid metabolism, so maybe this is going to fix getting that garbage cleared. And it regulates inflammation because your body responds to stress with inflammatory markers and says something's wrong. I'm happy to tell you that actually it's with Ocugen, is the company that sponsored my work. It's called OCU410, and we tested it in this model, ABCA4. It's associated with Stargardt's, but it has those AMD phenotypes. And it has now been approved by FDA to start clinical trials, both AMD. And where is it hitting? Remember that figure I showed you where all the different pathways? And you don't have to go, we're not going to go through all this data for time's sake, but it's hitting every one of those. We're not just hitting one pathway, we're able to hit multiple pathways. So I'm really excited to see how the trial goes, and hopefully our work, Apellis' work, Estella's, and all of you guys are going to come up with these exciting things, and we're going to help find treatments and cures for these debilitating diseases. Now I'm going to spend just a minute to talk to you about the phase, because I've been through this front to back end right now. Typical phases for a gene therapy study, cell therapy, they're all kind of the same. You can spend 20 to 40 years, and you're very young, and that might sound really daunting, but it's accelerated. I love being in science because it's exponential. Every year, what was a new tool last year is obsolete because there's something even more faster, more efficient, more effective. But it took around 20 years to get to the phase where I have multiple trials out. This is largely funded by our government. Thank you, Congress, because without government funding discovery science, you'll never get to phase 1 through 3. Government is the major funder of this discovery science space. And my other plug I'll give you is we have the science, we have the technology, we have really intelligent people in our field. We need sufficient funds to bring us to 2023, because when I started, funding was at 40%. I want funding to be at 40% for you. Do that, and we can find treatments and cures. And we don't have to spend 20 years doing that. Now, phase 1, 2, 3 usually takes about 6 years and costs about $250 million. It is not a joke. Phase 3 usually costs the most, over $100 million right there. Success rate is 10%. We'll talk a second about why, I think. And the effective cost of one drug being approved, and I think this was from a congressional website I recently looked, over $2.5 billion to do this. So we really want our government to help us come up with the appropriate funds for all of you, for all of us, so that we can reach this milestone faster, and also so we can have innovative funds so that we can actually go from 10% success rate to 90% success rate. It's possible. It is, the limitation is not technology, and it's not your brilliant minds and my brilliant mind. We can do this. And this phase, going from discovery and pushing it to translation, that is where foundations and pharma have been essential to our science moving forward. And I think AMDF, because they have been essential to keeping my lab floating, essential to getting my crazy project where, hang on, I have something, and I know it's outside the box, but I'm telling you, this thing can work, and now we have it in a trial for dry AMD, because it's hitting multiple of those networks. So all of these things coming together are really important. I love that, when I started grad school, it was you either go to industry or academia, and there the twain shall meet. I love that we're sitting in a group where we have industry, where we have representation from foundation and government, and we're all going to come together, because it takes all of us coming together to actually effectively and efficiently design treatments and cures. So, in summary, AMD is a complex disease, very complex. There are genetic components from multiple perturbations, and they're unique to each of us. There's several clinical trials underway for dry AMD and wet AMD, and there are 2 novel dry AMD treatments that are FDA approved. That is so exciting and new to see, and we just got approval for the therapy I developed to start for Stargardt's and dry AMD. Thank you, and I'm happy to take questions.
Questions off the top? Yep, go ahead.
Did you introduce mutations?
Nope, no. I put my genetics hat on. I saw this mouse, and I just bred and was looking at how many were affected. Okay, so and then I did what a geneticist does. We do a genome scan. I took 400 of them. Only 13% affected. This is not Mendelian. It's not a single gene. The mouse, by the way, had been around for over 20 years. It had been sequenced. It had been looked at. But everybody was looking at it with this very tunnel vision. So one other thing I'm going to say to you amazing young scientists is don't have tunnel vision in science. Let the data be the data, and let the data guide you. Because when you look at the data, you'll say, oh, this is not one gene. This is multiple genes. And there are probably other factors influencing those. Yes?
Are there any other therapies other than gene therapy for AMD?
Yes. In Kapil's work, there's cell transplant. therapeutics. That's a region, and I put it in that one slide, to say what was new at ARVO that I noticed. But yes, there'll be cell therapies. There's therapeutics. There's going to be device-driven. There's going to be therapeutics that are for different aspects of the disease at different stages of the disease.
We'll have a chance for some more questions in just a minute, but I want to give Mark a chance to come up here.
Yes. You're going to love his talk.
So I'm going to introduce Mark Roser, who, like I said in the beginning, is a patient who is living with macular degeneration, and he's going to come and share his story. He came down from Connecticut, and look forward to hearing a little bit more about your experience.
Great, Dan. Thank you very much. Neena, thank you very much, and I didn't applaud you yet, but thank you, Neena. The science, I think, is also just applause to all the scientists here, too, because I'm going to be talking to you today about three things, really: anxiety, self-efficacy, and opportunity, and all those really revolve around hope. And thank you for bringing the hope today, and the rest of you for bringing the hope today. I'll start off with some context, and the context is way back. I started getting my vision problems back in my early 20s, and some research from back then was from Dr. Brown and Dr. Brown, who worked together to look at the emotional, psychological impacts of different. And when they looked at vision loss, they did a simple test, which was they asked people, if you could look ahead how many more years you would live, how many of those years would you give up if you could get rid of your disease? And when they compared macular degeneration to diseases such as end-stage cancer, it was surprising. They found out people with late-stage macular degeneration would get more of their remaining years of life than people with end-stage cancer. And thank you to Matthew and to the AMDF for presenting some of the facts at the back of your fact sheet about fear. And so there's naturally a fear of vision loss, but for people who are in it, it continues. And so I'll just talk to you a little bit about that portion from anxiety. And when my symptoms started, the very first time I noticed it, I was at the New Haven, Connecticut, because they had the best selection of audiobooks for my commute back in the 90s and my cassettes, for those of you who remember cassettes. And so I was waiting to check out, and there was Venetian blinds above the checkout counter, and I noticed that one of them was dented, and you saw Neena presented the Amsler grid, and you saw that there were some squiggles. That's what it was starting to look like. And so I said, darn it, I must have dented my contact lens. And so, of course, I went home and cleaned my contact lens, and the next day it happened, and of course I did what any normal 20-something would do, which was promptly forget it and keep on going on with life. And of course, by the good grace of my father having a conversation with me a week or two later, he said, no, you should probably get that checked out with, don't mess with your eyes. So I went in, made an appointment with the optometrist, and it took the 6 to 7 weeks to get the appointment with the optometrist to have this issue looked at. Got into the optometrist, and of course the optometrist didn't have an Amsler grid, didn't have tests available to test for this, so he took out a ballpoint pen and drew some straight lines up and down and had me look. Well, that's something that you should have looked at by a specialist. That's not something I can take care of here. And so he referred me to a retina specialist. And so, of course, like any 20-something, I didn't jump right on it. I waited, and of course, thanks to my father, I was able to get some inspiration and a little bit of courage, and then go in, get an appointment. 5 weeks to get in and looked at, and said, no, you have a idiopathic or unknown or juvenile form similar to macular degeneration at the back of your retina, and we have a treatment for that, which is surgery, and it's laser surgery. And so I'll give you an analogy now. So what I like to explain with wet macular degeneration, if you can imagine walking down a sidewalk and you see a weed growing up through the sidewalk or weed growing up through the pavement, you'll start to crack and get brittle. It loses its stability. That's what happens when you have a new blood vessel. It grows up the pavement and eventually it cracks and the bright minds of the day said if we took a very intense heat, we could seal this over in service of keeping it from getting worse and spreading. And so with laser treatment, a very skilled surgeon would very microscopically take the laser and just about 250 to 400 bursts of short laser, they'd seal off the area, and you'd have the weed burned to smithereens and the pavement around it sealed up, so that way it wouldn't come back. And so problem solved. I was grateful, and I go home. I was in the process then. I was an engineer at the time. I was actually enlisting in the Navy, and after working in designing hydroturbines and finding it boring, got in to do the exam and was not qualified. And I said, what's going on? I thought I had this taken care of. I couldn't pass the vision test. And so then I went back into the doctor. Turns out it was getting worse. More surgery, and now I start to get a little bit more concerned and nervous. And so I try to figure out, how do I measure my vision at home? How do I tell if it's worse? Because he said, now you're good to go. You should be okay. See me in 6 months. So I started to develop my own tools at home to check on my vision. And so the way I did that was I learned over time that the brain fills in things. So if I look at a yellow wall, even though I have missing spots in my vision, the brain fills in and makes it yellow, so it's hard to tell. I can't tell if my vision's getting worse just by looking at a surface. So I came up with my own tools and tricks. I used dashed lines, for example, because the brain can't estimate dashed lines. I understood certain bright surfaces the brain can't keep up, and they'll start to show up as a instead of a just of a neutral area. And so over time I learned that if I had a blind spot and I could position myself just right underneath a light, and I could find it where it's just framed just right, I could go sit in that chair and look at it every day. And if it no longer fit in that frame, I knew something was wrong, and I could go in to see the doctor. And so that's how I would be able to learn, because I knew that the sooner I got into the doctor, the less laser surgery they'd have to do. And when I walked out of the office, I'd keep more of my remaining vision. And so that is a technique I started to use and develop. And when it happened, the next time I walked in and said I need help, and he said, I can't see anything. The instruments back at the time couldn't measure it. So I had to wait a couple weeks, and a couple weeks later I complained, went back in again, and was able to get treated. So I tell you this story to kind of show you how it led up, but then going back to the anxiety part, so what this meant in my life was that as soon as I had a treatment and was able to get stable, you go through a certain period of mental adjustment and coming to terms with things. And it's difficult because you're losing, for me it was losing binocular vision and depth perception. And so things I could do before I was having trouble with, just putting a cup down, I'd crash things down as an example. You break glasses on the counters and embarrassing things like that. And so just that sensation of not knowing, because then as it progresses, then it happens again, and then you get treated again, and you have to come to terms again. And so it's that progression, and when you hear diseases, think of these words, progressive eye disease, link that back to what that is internally for somebody, to be able to get hope and then how to lose the hope, to get hope and to lose the hope. And that's what becomes demoralizing. And so, for me, how it showed up is I'd sleep with a light on, because I'd go getting up in the moonlight in the dark, I wasn't sure if it got worse. So I'd want some light on just so I could cover one eye and go, oh, okay. And so it's just, it can be, it's frustrating, because I was told that, for people in that condition, my doctor said to me, it's roughly 10 to 16% year for it to start going on the other eye. So, think 6 years or so before you lose it in the other eye. And so it's those types of things where I said, and I just applauded the scientists here just for the hope side, because now what we've replaced that with was the first anti-VEGF, which is the treatment that allows, as the plant grows up through the pavement, for a neovascular, for a wet AMD for targets. Same thing for diabetes and proliferative diabetic retinopathy. It's basically a weed killer, so the weed shrivels. and it no longer grows. And so if I had that, I would still have my vision today. After multiple surgeries, they eventually lasered over pretty much my whole central vision. Said, okay, you're legally blind now, we're stopping treatment. So that's how it ended up, and so I say for the treatments there, so that's that. Number one is the treatments that we had, number one, are phenomenal now, 92% effective as a weed killer of stopping the weed growth. But what I learned, and I was working as a consultant in Pfizer to the team that was developing the first Macugen, and told them about my tools I designed at home, and they encouraged me to move forward with it. But what we found is that even though these tools are now available, these drugs are now available, 90+% effective, macular degeneration, diabetic retinopathy are still the leading causes of vision loss. So why is it that we have drugs that are 90+% effective, yet people are still losing their vision over going legally blind? And a lot of it is just because of doubt, fear, and worry, the anxiety, and a difficulty in wanting to get to the office quick enough. Through the support of AMDF, we did some additional work. I did some looking into what's the average time it takes somebody to get in to see their doctor for macular degeneration, and it was on the vision chart that you look at the doctor's office. It was 4 lines down before somebody would raise their hand. So if you look at their normal annual visit to the visit when they got yellow flagged and sent to the doctor, 4.2 lines of vision before people are willing to raise their hand. So it's basically, I won't go in and ask for help until I can either no longer drive or have difficulty reading. And so things like that are things that I'm passionate about my side. The things that the scientists are here that are passionate about are changing the way in which we treat things, and so which we can prevent those weeds from going in the first place, that we can stop that proliferation so that the retina stays intact. So that way we also have cellular features of the amyloid plaques so that we can prevent them from separating, so that way we are able to stop the disease. It's life-saving that we're able to keep it from getting worse. But in the future, what the promise is that we'll prevent it from getting bad in the first place. And that's, as I said, that's necessary because right now, even if we have these phenomenal treatments, 90% effective isn't good enough because human nature is human nature. So I'll just close with, so that was my story of anxiety, and then self-efficacy is really the opposite of that. The willingness to see a problem and go get help is what is known as self-efficacy. And so I think there's a tremendous amount of opportunity there as well. And through the work and support of AMDF, and through the support of my friends at Pfizer, they encouraged me to take some of the tools I developed in my own home use, which is basically, we now give away tens of thousands of free vision tests a year that use these tools that help people with simple tools. I can go over them later with anybody interested. But basically, the whole idea is we're trying to give people some power at home between their annual eye exams to check their vision. So if they see a problem, they're willing to go in and ask for help about it. And we're also developing new outreach tools. So if people aren't even in to see the doctor yet, and then they get encouragement and they're willing to step up and say, I should probably go and see an optometrist, I should see an ophthalmologist, I should get myself into the system so that I can get help. So to me, transforming from anxiety to self-efficacy and hope and the opportunity is what today is about, and I'm just grateful that I was invited here, and thank you so much, Dan and the Alliance. I appreciate you. Thank you.
So that concludes the presentation portion of the briefing. I do want to emphasize that AMD is a really unique condition in that losing that central vision is fundamental to how we experience the world. That central vision, imagine looking at a family member's face or things like that and not being able to see their expression, not being able to understand the way that you're interacting with the world in the same way. And then that progression of the disease as it gets to more advanced stages can be quite significant. So I do want to thank again our sponsors and our supporters for helping bring today's event to fruition. We do have about 10 or 15 minutes left for some Q&A, but also we've got the experience option in the back, opportunity in the back. And so I would encourage congressional staffers who are here to please go ahead and head to the back now. I'd love for you to experience what AMD looks like from a patient perspective. It really helps bring home what people experience and why research is so important. If you have questions, our speakers will be here. We have some lunches in the back for folks and some materials in the back from AMDF. And again, thank you AMDF for the lunch, but please take some time and experience what patients are truly seeing with AMD. We'd really appreciate the opportunity to share that with you. So thank you very much.